Hi, ladies and gentlemen. I wanted to walk you through how to do the second part of the protein synthesis practice problems. Okay, so we're going to start here on slide 5. This is an example for you guys. Okay, and in looking at the example, if you guys take a look at the messenger RNA, okay, the messenger RNA has the codons. Okay, these codons, okay, remember codons are 3 bases long. Okay, those codons, okay, are going to code for a particular amino acid. Let me move that back, sorry. Okay, so if I go to my first codon here, that first codon, GGA, I'm going to use this codon chart that's at the left. And that codon chart, let me make it a little larger here so you guys can see a little better. I am looking for this sequence, GGA. G, so I start in the middle and I work my way out. First base of the codon, second base of the codon is also a G, third base of the codon is an A. So GGA is going to code for the amino acid glycine, GLY. The second codon, if I go to the second codon, it's GAA. I come to my codon chart. I find the G. The second base is an A, so I'm looking at these 4, right? So I'm working in this quadrant. My second base is an A, and my third base is an A. So I move further out, in this outer ring. So I've got G, A, A. And when I look at the amino acid that's coded for by that sequence, I get GLU. So you'll see that on my transfer RNA, I've changed the first amino acid to glycine, the second amino acid to glutamate. Take a look at the third codon. The third codon is AUG. If you take a look at your codon chart, AUG, that codes for, it's kind of cut off here in the picture right now, that codes for methionine. Please check your slide here, because originally this amino acid was incorrect. So that should be methionine, MET. That is our start codon. Then you move to the next 3, UGU. I find UGU. That codes for the amino acid cysteine, CYS. And then the last codon is CGG, last codon is GCC. And that's a common mistake, is using transfer RNA instead of using the messenger RNA to figure out what amino acid. So GCC, if you take a look at it, GCC, that codes for alanine, ALA, which I spelled ALA here. So let me fix that. So that's how you go about completing these protein synthesis practice problems. So if I come to, let me fix that. When I come to slide 6 and I'm working my way through this, right, you've already figured out the messenger RNA transcript, right? So transcription happens first in the nucleus. I'm going to look at this first codon. It's UCU. I'm going to look at my codon chart. UCU, that codes for serine. So the first amino acid in my sequence, and again, I can just click on this bubble. The text box is already there. I'm going to type in serine. My second codon is UGU. I come back to my codon chart. UGU codes for cysteine. So again, I click on my bubble, type in CYS for cysteine. And you're going to continue that process for the rest of the codons. Once you have the amino acid figured out, the transfer RNA that brings that amino acid to the messenger RNA is complementary to the messenger RNA. So once you've completed your amino acid sequence, you're going to go back through and I'm going to say, okay, what's the complementary base to the messenger RNA? Well, this is a U, so complementary to U would be A. Okay, I click on my next bubble. Complementary to C is G. Complementary to U is A. Okay, so that's my anticodon that's on my transfer RNA that will bring the correct amino acid, in this case it's serine, SER, to this messenger RNA. And then eventually these amino acids will be linked together to form our long chain of amino acids that will then fold into proteins. Okay, again, my next codon is UGU. So that transfer RNA that's complementary would be A, C, A. And again, if I've forgotten what bases pair together, this is messenger RNA to transfer RNA. I can go back to slide 3. We are now looking at the base pairing rules that are in red on slide 3. Messenger RNA is on the left. Transfer RNA is on the right. Adenine pairs with uracil. Uracil pairs with adenine. Guanine pairs with cytosine. Cytosine pairs with guanine. The key thing is we are talking about an RNA, so there are no thymines involved. All right, ladies and gentlemen, I hope that gives you a little bit of an example of how to complete part 2 of this assignment. So part 1, we were doing transcription, DNA being converted to mRNA. In part 2, we are taking that mRNA, using it to determine what amino acid sequence first. Okay, we're using the codons, 3 bases long in the codon chart, and you're using your transfer RNA. Okay, and that transfer RNA is going to be complementary to the messenger RNA. All right, ladies and gentlemen, have a great day.